Assalamu alaikum. So today our the topic of discussion is protein catabolism. So in this video may be somehow large, but I will complete the whole protein metabolism here. Okay. So protein catabolism, we are gonna study how protein are degraded first into amino acid, because we know that proteins are made up of amino acid. So when there is a degradation of proteins, it produce amino acid. Okay. So the first thing we study, protein are degraded in amino acid. Then how amino acid are degraded into different compounds. Okay. So first, in the further degradation of proteins, we should know that protein degradation has two method by which we can degrade the proteins. Okay. The first method is called lysosome pathway. Okay. Lysosomal pathway. Okay. This pathway, which is lysosomal, it is ATP independent. Okay. You should know that there is no need of ATP here. Okay. So we say it ATP independent. Then the second pathway by which we can degrade protein into amino acid, it is called ubiquitin proteasome pathway. Ubiquitin proteasome pathway, which is ATP dependent. Okay. You just need this thing. Okay. Don't go too deep that how this ubiquitin pathway is important and how this occur. You just need lysosomal pathway, in which the lysosome is the organelle which is used and cause the protein degradation into amino acid. Here ATP are not used. Here the enzyme is used, which is mostly called hydrolase enzyme, acid hydrolase, etc. Okay. Hydrolases are used here. Okay. These are used here, hydrolases. Then we have the other pathway in which the ubiquitin and proteasome are used for the degradation of protein into amino acid. And you should remember that ubiquitin and proteasome is not an organelle, it is a macromolecule. Okay. Which causes the degradation of protein into amino acid. Okay. And ubiquitin is just attached to specific protein. Okay. If these are two protein, okay, these are two protein, and ubiquitin is added only to this protein, when this protein is ubiquinated, this protein gives signal to the proteasome that I am ubiquinated and you will degrade me. Okay. So it means ubiquitin, whenever is attached to any protein, this is like a tag. When this ubiquitin is tagged to any protein, it gives the message to proteasome that this protein want to be degraded. Okay. When any protein are not ubiquinated, it means when ubiquitin is not added to any protein, this will not be degraded by the proteasome macromolecule. Okay. So proteasome only degrade those protein which are ubiquinated. Okay. So this process gives us very specific method by which specific protein which are ubiquinated are degraded. So when we degrade the protein, these are converted into amino acid. So now by the degradation we get the amino acids. Okay. So further we degrade these amino acid. Okay. And we will get the ammonia NH3, which is the mostly end product of amino acid. Okay. So how NH3 is produced? We have two pathways for the degradation of amino acid. First pathway is the transamination. Why? Because the deamination is the process by which ammonia is formed. Okay. Why we need transamination, I'll tell you in the end of this. First let me tell you, in the transamination we have mostly alanine which is converted into pyruvate. Okay. Which is converted into, very good, pyruvate. And which enzyme is used here? Look, this step is called transamination. So enzyme should be transaminase. This enzyme is called alanine transaminase. Okay. So second thing, we have aspartate, which is an amino acid. Like alanine is an amino acid, aspartate is converted into oxaloacetate. Very good, oxaloacetate. So here the enzyme should be transaminase again, but here that one, the alanine transaminase, here should be the aspartate transaminase. But let me tell you one thing, transamination is a process in which amino acid is converted into keto acid and keto acid is converted into amino acid. So what does it mean? Okay. I in the lecture, previous lecture, I told you about the TCA cycle. This was the TCA cycle. If we want to draw this TCA cycle in a simple form, I can draw like this. Pyruvate is converted into acetyl CoA, then acetyl CoA is converted into, very good, into straight, and then straight is converted into acetate, then alpha ketoglutarate. The alpha ketoglutarate is converted to succinyl CoA and succinate, okay, and succinate. I just write succinyl CoA because succinyl CoA and succinate are almost same thing, okay, which is converted into fumarate, which is converted into malate, and then oxaloacetate, okay. This was the whole TC cycle. You can see my previous video if you want to do this TC cycle, okay. So why I am telling you this TCA cycle? Look, in the TCA cycle, these whole the compound intermediate which are present in the TCA cycle, these are the compound which we normally call the keto acid. These are the keto acids. And what are amino acid? You know, amino acid are the compound which form the proteins, which are, we know, alanine, methionine, and different aspartate, glutamic acid, okay. These all are the amino acid. So I was saying that transamination is a process in which the amino acid is converted into keto acid and keto acid into amino acid. I will show you how this definition is. First, you should remember that when alanine is converted into pyruvate, okay, this alanine is a amino acid, and pyruvate is a keto acid. So amino acid is converted into keto acid, but the second line was definition was, and keto acid is converted to amino acid. How? Look, we have alpha ketoglutarate, which is present in the TC cycle, alpha ketoglutarate. So alpha ketoglutarate is a keto acid, and it is converted into glutamate. Like this, we have in this reaction was this reaction is also same. Alpha ketoglutarate is converted into glutamate. Okay, so in both reactions we have alpha ketoglutarate which is converted into glutamate, but the difference is amino acid and keto acid which are produced. Okay, so look, in this reaction definition was transformation was the reaction in which amino acid is converted into keto acid. Look, amino acid is alanine is converted into keto acid, and keto acid is converted to amino acid. Alpha ketoglutarate is a keto acid, and glutamate is a amino acid. So keto acid is converted to amino acid, and amino acid is converted to keto acid. Like this, we have the same reaction. Okay. So why we do transformation? Have you checked one thing, that whenever we have transformation, glutamate is always formed. Here the glutamate is formed in the transformation reaction. Here the glutamate is formed. So that is the reason that we want transformation, because we want glutamate. For the glutamate, we need glutamate, and why we need glutamate? There is a reason, because deamination is a process in which the NH3 is released, in which the NH3 is released, in which the ammonia is released, and glutamate is the amino acid which release this NH3. Aspartate does not directly release NH3. Alanine does not directly release NH3. So we just want these amino acid first to convert into glutamate, aspartate to glutamate, so that these amino acid NH3 is become the part of this glutamate. When glutamate, we get glutamate from these transamination, then glutamate, here come glutamate, and this glutamate is converted into NH3, but at the same time it forms alpha ketoglutarate. Okay, so just repeat this thing. We want transamination because we want glutamate. When we get glutamate, we deaminate it to get NH3. And what is the fate of NH3? What is this NH3 will go? This NH3 will go to urea cycle and produce urea. Okay, so and produce urea. And what will alpha ketoglutarate? This is again used. Okay, this is again used in transamination reaction, and it can also go into the TCA cycle. Okay, because it is a part of TCA cycle. Okay, so it can go to the TCA cycle, and it can become the part of reaction in which transamination it is used. Okay, so this has many use, and NH3 is just waste. It just go to the liver, liver in which the urea cycle is present and it is converted into the urea cycle. So in this lecture we know that proteins are converted into amino acid by the lysosomal and ubiquitin pathway. Then we have amino acid eater. These are degraded into first these are transaminated to glutamate, then these are degraded to NH3, which is converted into urea by the urea cycle. And I have a video on the urea cycle which is separate video. Okay, so we have urea. Okay, now I want to tell you something more, and that's, look, in this reaction we convert alanine and we convert glutamate into alpha ketoglutarate. Okay, glutamate is a amino acid. Okay, amino acid, and alpha ketoglutarate is a keto acid. Okay, keto acid, what you, if you are thinking what are the keto acid, what are the keto acid, you just remember these, the intermediate in the TCA cycle, these are called the keto acids. Okay, don't confuse yourself, just make something easy. So some question are most, not some question, these questions are mostly asked in the MCQs, that which amino acid produce which keto acid. Like the, if we have methionine which will produce which keto acid. Like in the TCA cycle we have pyruvate, I am again dying the TCA cycle, which is converted into acetyl CoA, which is converted into state and converted into acetate and alpha ketoglutarate. Okay, just write the main thing, which is converted into succinyl CoA and succinate. Okay, succinyl CoA and succinate, which is converted into fumarate, and which is converted to malate and then oxalostate. So these are called the keto acids. Okay, these all are called the keto acid. Okay, which are used in the TCA cycle and also pyruvate and acetyl CoA. Okay, so in this section we are going to study which amino acid produce pyruvate, which amino acid will produce acetyl CoA, which amino acid will produce oxalostate, which amino acid are going to produce fumarate. So we have mnemonic, we can easily remember that which amino acid is going to form pyruvate and acetyl CoA, because we have a lot of amino acid and we have lot of keto acid. So for normal person to remember the which amino acid is converted into which keto acid is very difficult. So if we have mnemonic, we can easily remember. So first of all, we are going to do which amino acid are going to produce pyruvate. So I have a mnemonic for this pyruvate. Look, the mnemonic is: a great soldier cut their timeline. What does it mean, a great soldier cut their timeline? Timeline is a pathway by which the different food, materials and weapons are supplied from the main military source to the area where the war is present. Okay, there the soldiers are present, and the path by which the food is transported and all the weapons are transported, this is called the timeline. Okay, in the army this is called the timeline. So if a person want to defeat this army, he just need to cut this timeline. When the timeline is cut, there is no supply of food and this army will be soon die, and the person will win this army. Okay, so we have the simple: a great soldier cut their timeline. Okay, a great soldier cut their timeline. This mnemonic is for the pyruvate. What does it mean? This is means that alanine. Okay, alanine produce the pyruvate. So means alanine. Then G for glutamate. Then S for very good, serine. And C for cysteine and cysteine both. Okay, and we have T and T. So first T is for threonine and tryptophan. Second T is for tryptophan. So these are the amino acid, okay, which are converted into pyruvate. So you can easily remember by this mnemonic. Okay, so the second mnemonic is Ali has great practice. I am very tired. Please take Ali. This mnemonic is used if some person say please if some person come to me and say, Dr. Easy, please do this work for me, and I just reply, oh, I am very tired, please go to Ali has practice in this work, he will do your work. So the mnemonic is like that: Ali has great practice, I am very tired, please take Ali with you to do some work. Okay, so this mnemonic, the Ali has great practice, this line is for the alpha ketoglutarate. I always say that kg is king. Why? Because everything is start from the kg. So if you want the which amino acid are going to produce kg, so you has amino acid, this line gives you the amino acid for the kg. This kg is produced by Ali has great practice. So Ali means arginine, as for histidine, I'm confused, it is arginine or alanine. Okay, let it confirm. Okay, as for histidine, G for glutamate, and P for proline. Okay, so these are the four amino acids which are going to produce alpha ketoglutarate. This Ali has great practice. Then the second one, after the alpha ketoglutarate, we have succinyl CoA. So which amino acid are going to produce this succinyl CoA? So for the succinyl CoA, we have mnemonics I am very tired. So I for iso, leucine, M for very good, M for methionine, I am very, V for valine, and T for very good, T for threonine. Okay, so we are now going to fumarate. Okay, we done this. So fumarate, please take these two words for the fumarate. Okay, look, we've done the alpha ketoglutarate, we've done the succinyl CoA, then the next one is the fumarate. So next one line in the mnemonic is please take. So please, P for phenylalanine, and T for tyrosine. These are very important because phenylalanine normally can be converted into tyrosine. Okay, whenever you have phenylalanine, there should be tyrosine in the mnemonic, and in everywhere when you will have phenylalanine, you have tyrosine. Okay, remember this thing. Okay, so now we done this fumarate. Next one is oxaloacetate, and we have, remember, on in the mnemonic we have only one thing which is remaining. We done alpha. Ali has great practice for the alpha ketoglutarate. I am very tired for the succinyl CoA. Please take for the fumarate, and we just remaining with the Ali, and this Ali is for the oxaloacetate. A for aspartate. Okay, so we're done with this thing, but this pyruvate, which is a great social card, their timeline. A for very good. A for the alanine, we have glutamate. I make this mistake, so I want to clear this. This is glycine, G for glycine. Okay, then other things are the right. Okay, so this was the whole thing, which is called the conversion of glucogenic amino acids. These are all glucogenic amino acids because they are going to produce this intermediate of TCA cycle, and you know this TCA cycle is when convert back, it will produce the glucose. So these are all amino acids which are right here are called the glucogenic amino acid, and each keto acid has specific group of ketogenic amino acid, and you can remember these group of ketogenic amino acid, glucogenic amino acid by this mnemonic. Okay, so for this I think the video is very long, so we will see in next video. Till that, bye bye.